So here you got this chart, and I'm going to quickly sketch it. And you got this buffalo population. There's like these numbers. I'm just going to put like a couple numbers here. I'm going to put them in thousands. So here's 60,000. Here's 50,000. Here's 40,000. Here's 30,000. Here's 20,000. And here's 0. And here's 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. And it started off at 50,000. The population started off at 50,000, and then it went down after a couple of years to something like this. And I'm just going to kind of draw it. It's going to be a sketch, so deal with it. I don't want to hear that it's not exactly like the graph. So let's read it together, shall we? What do you think happened here? It went from 50,000 down to here. What happened? The virus killed a bunch, right? So this is a viral death right here. This is death by virus. It devastated the population, didn't it? Yeah. I mean, it almost killed them all off. It's actually much higher than this. It's not 20, but whatever. So then, after the virus, the ones that survived were the ones that were resistant to the virus. So now these guys are not good. This is called, by the way, you could also look at this as an example of evolution. Natural selection. It selected those beasts that were not resistant to the virus. So we're down to 20 grand, and so now these are resistant. Virus is not killing them anymore, but then they have a lot of land, a lot of food, a lot of water. A lot more than they used to have, right? Yeah. Compared to their population, rather. The amount's still the same, but there's less beasties to share that with. So now many more of their babies survive, don't they? Yeah. So they start to have more and more babies over the many years that follow, until they reach this point here. You think that that's the carrying capacity right here? Well, hold on. I'm okay with you making that guess. Let's say something happened here. What's starting to happen here now? They're running out of food. Oh, so what happens because they run out of food?
Some start to die. Like the most fit.
Well, whatever the most fit is, they're going to survive them. But there's going to be a lot of death. Some of it random, some of it because they're not fit. But they go down and they're dying, and then they keep dying. Oh, the poor little things. But at some point, the population goes. What happens is the plants start to grow back, right? And there's less of them, so now there's plenty more water and land. So what happens to them now? They start going up again until something happens. What happens? They start to run out again, and the same thing starts again, right? So then they go down, and they go again low, and what do you think is going to happen after that? It's going to come up again. So where is this going up and down around? What number is it going up and down around? It's 60, right? It goes up and down around here. You can see it goes up and down, right? It goes up and down around 60. Let's go back to the original picture, which is a better drawing. Again, if you have problems, look with the handout. Look at this. There's a devastation. It starts to increase. It goes too high. Remember, there's a lag. It takes time for things to starve to death, right? There's competition. So it's not like they reached the carrying capacity and they died. No, they reach the carrying capacity. Everybody's starving, fighting, whatever. They come down, they're dying, and they go back up. So they start to die off. They go all the way down here, and then what happens to them? They jump back up. So where are they going up around? 60,000. So the answer is 60,000. All right. So this is what somebody guessed. Let's see if we can do it together. This one's easy. You see these, you should be very happy. Here's where people mess up. They see this, they get too happy. I got this, done. Next one, they got the wrong order. They mixed it up. They reversed it. They didn't pay attention to the pluses and minuses. They didn't think about it. They didn't check their answer. And because they didn't do all that, just a couple mistakes and you're done. Just lost a really good opportunity. These are the best ones. This is one of those questions where you want to make sure you get it right because it's easy. So when you're reading the cladogram, if you see the trait here, that means the stuff that came before it did not have that trait. So in this particular trait is a trait that these species have that green algae do not have. And since chloroplast, everything here on this list has a chloroplast, number one is not a possible choice. Does everybody agree with that analysis? Yes. All right, so then what is the only trait that mosses, ferns, conifers, monocots, and dicots do have that green algae does not? It's 2, so 2 goes there. What is the only trait that green and mosses don't have that ferns, conifers, monocots, and dicots do have? Vascular tissue. Look, negative, negative. Only green algae and moss. Now, on the state exam, maybe they reverse it, right? Maybe they put the species on the left and they put the traits on the top. Maybe they reverse the chart so it's facing the other way. See, they can do all kinds of things that kind of throw you off. So that's why it's important you take your time. Don't try to memorize this question. Just remember, this is what you need to know, what you need to learn is the logic, the fundamental problem solving beneath the details. All right, which is the only trait that conifers, monocots, and dicots do have that ferns, mosses, and green algae do not?
Seeds.
Seeds, you see? They have it, these 3 don't. So seeds, 4. And what's the only trait that only monocots and dicots have? Flowers, number 3. And that's that easy, you just got an A on that question. Moving on. All right, so you see you can move these around any kind of percentages you want. It's real easy. Did you guys do this one?
No.
All right, so go try to do it. See if you can do it. So let me zoom in a little bit. What did you say? So now let's continue reading. It says, students conduct an experiment to test whether or not the trait for pollen shape and trait for flower color are next to each other on the same chromosome in the pea plant. They cross 2 plants that are known to be heterozygous. So this, number 1, they're heterozygous. That's important to know. So that means that mother and father, or the both plants are heterozygous. So that means that both plants are big L, little l, right? And big P, little p is crossed with a big L, little l, big P, little p. Do you agree that that's what I just read? Okay, so that I read that. Okay. Long pollen shape is L. So the other thing we know, look at what I'm doing. I'm writing down what I know. You need to do that on your when you're taking tests. So big L, so big L is long pollen shape, and little l is round pollen shape. And big P is purple. Now you wouldn't have to write all this down if you were taking a test because it would be in front of you on the test. But you might want to make sure that you know it's heterozygous. You might actually want to write this. See, they didn't draw this out, did they, in the problem? But you might want to actually go ahead and do this part. But this part that I'm doing right now is already done for you. And this is red. And this is flower color, obviously. So that we know. Then it says, click on the value of each phenotype frequency on the graph that would be expected if the traits are linked and the dominant traits are next to each other on the same chromosomes for both plants. Okay, geez, that's a lot, right? So now we got to go back. Now you got to, somebody had to have taught you what does it mean to be heterozygous. You have to have known where are these genes in a chromosome. So you have to know that you have a heterozygous, both of these parents have two chromosomes, right? One from their mother and one from their father. And the two genes, let's say says the two dominant genes are next to each other. Let's see if the other one is blue. And it's a big P and a big L. And what is the other? What are the genes from on their other chromosome? Little L and what? And little p. How do I know it's little L and little p? Because both parents are what? Heterozygous. If they have a big L and big P on this chromosome, they have to have a little L and little p on this chromosome because this is the only place you're going to find it. So just by going through all that work of trying to figure out what a chromosome is and what a gene is, you have already an advantage with this problem. Okay, so that was important to know. So all that was just trying to write down what you know. All right. Now question, if you had a normal heterozygous cross, if they weren't linked, so there's two possibilities. They were not linked, how would you figure out what kinds of phenotypes you would have? What would you do? You'd have to know the gametes, right? So you have to do a dihybrid cross. And you know how to do that, right? You have to do a dihybrid cross, so you have to do FOIL, wouldn't you? You'd have to go big L goes with big P, that gives you big L, big P, right? And then you'd have to do big L goes with little p, so it gives you big L, little p. And then you'd have to do little L goes with big P, so it gives you a little L, big P. And then you have to do little L, little p, and then you'd have little L, little p. And of course, you would have the same gametes for the other parent because the genotype is the same. So you have big L, big P, big L, p, little L, big P, little L, little p, and then you do your columns and your rows, and you'd fill them in, right? And then you'd count. Now you could spend 5 to 10 minutes on a 90-minute exam. You can spend 10 minutes out of 90 is 1/9 of your test time on this one problem to figure out how many different gametes, how many different embryos how many different zygotes are going to be different long or round and purple or red, right? You could do that, but you really shouldn't. What you should do, though, which should be something that you could do easily, is you should remember 9 to 3 to 3 to 1. You should remember that there are going to be 9 big L blank, big P blank. 9 of them. You should remember that there's going to be 3 big L, right? Big L. Big L, and then with blank with a little p, and you're going to have 3 that are going to be little l, with some kind of big P blank. And you're only going to have one of those in ratios, that's going to be little l, little p. That's something you should know. 9 to 3 to 3 to 1. If you had a regular cross, and it was just, let me just go and make this short because I'll leave less room. If you had just a monohybrid cross, where you had big L crossed with big L, little l, you should know the other ratio. What's the other ratio you should know? 1/4 big L, 2/4 or 1/2 big L, little l. So half are going to be heterozygous, one quarter are going to be homozygous dominant, and one quarter are going to be homozygous recessive. You should know that ratio as well. All the other ratios you can figure out as you go. Is that okay? So if you knew this ratio here, you wouldn't need to do all this math. But the problem is that these two genes are linked. So then that changes the dynamics of the problem.
Can I borrow 10 Chromebooks? Because I usually borrow from Polygon. She's using the same one.
Yeah.
I apologize. I don't.
Here. Here you go. All right. So the problem is that these genes are linked. That means you're not going to get a random, this is not going to happen. Because you're not going to get them mixed up. The whole reason you get these four possibilities is because crossing over can happen, right? And you could take the whole cart with you, just bring it back. It'd be great. So when you're looking at this, you have crossing over happening, and that's why you end up with four possibilities on each parent. But if crossing over is not happening, which is what's happening when they are linked, right? Remember, when they're linked, that means crossing over is not likely. So they are linked. What that means is that they're not going to move like this. There are not four possibilities. There's only two possibilities. What are they? You're either going to get number one or you're going to get number two. That's all you get for both parents. So now you're either going to get big L, big P, or you're going to get what? Little l, little p. And that's for both parents. Big L, big P, or little l, little p. That's all you're going to get. So now what does that turn into? That turns into big L, big L, big P, big L, big P, little p, big L, big P, and little l, little p, little p. Well, what do these three look like? This one, and this one. They all have a big L. They all have a big P. Do you see that? And because they all have a big L and big P, what are they all? So 3/4 or 75% are what? Long and purple. And then 1/4 or 25% are what? Round and red. And so this was it. This was the answer that you needed to write down. Now let's say you weren't sure. Let's say you forgot about linked genes, or you're confused. Somebody help me out. Darla, Onisha, grab your things and go to the office. Tell them you can't stay awake. We're doing a review. You can't stay awake. You need to go and take care of yourself. Go to the office, please. I'll write up a referral later and hand it to Ms. Perez. Just go. So when we switch back for a second and we look at this, if you try to do the first one, like you can't remember, what's 9 out of 16? Somebody take out a calculator and do the math. Notice there's no calculator here because you don't have to use a calculator. But you know 9 out of 16, is it? No, Onisha, you got to go to the office. Please, just tell them you're sleeping in class and Mr. Mendoza said I have to go to the office. He's going to write a referral. Please do that for me, please. Oh, okay, I'm sorry. I thought you were not moving. All right, so 9 out of 16, who can do the math real quick? What is it? 0.56, right? Can I do 0.56 on this? That's a clue. It's a clue. You can't do 0.56. You can't do 56. Can you do 56? Look. It's either 50, it's what? It's 50 or what? Is that 50? I don't even know what number that is. I don't care. It's not 56 though, right? But you can't do it. Okay, let's say it's 60. It doesn't matter. It's not 56, right? So you can't do it. That's a clue. That's a clue, guys. So the fact that you can do 50, can't do it, you know that that's not the answer. So it's not a regular 9:3 or 3:1 ratio. So if you tried the other way, if you did linked and you realize it's a 75-25, can you do 75-25? Sure, I can do 75 purple flowers, long pollen, and then 25 red flowers and round, long pollen and purple flowers and red flowers and round pollen. Sorry about that. And you can see that, let's check it and score the item. What do we get? All right, so it is what we thought it was. And it's just simply, is the gene linked or not? If it's not linked, this 9-3 to 3-1, and you don't have to do a dihybrid cross, but you know it would be a dihybrid cross, you'd use foil and all that if you have to do it. And if it is linked, then you know it's not going to be that. And you could easily do a quick dihybrid cross with only 2, or you memorize 75-25, whatever you want to do. I like to work problems out. Are we all good with that now? So that's recorded, and I'll try to forward that, because I know some people were absent. So now a scientist isolates a number of non-photosynthetic prokaryotes. What does that tell you right away? What are the things we know? We're going to write down what we know. Remember, write down what we know. What does that mean, non-photosynthetic? That means what? What does that tell you right away? No chloroplasts, right? Because if they had chloroplasts, they would do photosynthesis. But, okay, they don't do photosynthesis, so that also tells me they're what? It tells me they're what? It tells me they're not plants. I heard someone say that. All that's true. All this is stuff I know just by that word, non-photosynthetic. And then they say prokaryote. What does that tell me? They make their own food. They're autotrophs. I'm sorry. Non-photosynthetic means they're not autotrophs. They don't make their own. They're heterotrophs. So prokaryote. What does prokaryote mean? No nucleus. There are no membrane-bound organelles. What else do you know? What do we call those kinds of prokaryotes? What do we call them? They're either bacteria or archaebacteria. But bottom line, they're not eukaryotes, right? So they're not fungi, they're not plants, they're not animals, they're not protists. They're prokaryotes, they're bacteria, archaebacteria. Okay, so we knew all that. Now life should be easy. Let's just apply it. Which structure would be, so what I would do if I were you is I would highlight, I would go over here, I don't know if you can in this thing. You don't have to give a mask. It says would be, oh, okay, forget it. So you can just highlight would be means yes, found in these cells that are prokaryotes and non-photosynthetic. So it turns out you can go through this thing and you can do strike through, right? Would they have cell walls if they're prokaryotes? Yes, they would. That's one of the characteristics of bacteria. Bacteria, that's the other thing that you knew, is that bacteria have cell walls. All right. So would be. But let's assume you don't know that. Let's assume you don't know that. Or let's say, yeah, man, I don't know. I don't remember if they have cell walls or not. Okay, let's go on with the other. Could they have chloroplasts? No, in fact, we already said they don't have chloroplasts. Could they have Golgi bodies? No, because they're membrane-bound. Golgi bodies, as you know, are membrane-bound organelles. And do they have a nucleus? No, that's the definition of prokaryote. So if you said they don't have cell walls, then you're stuck, because then none of these are correct, right? But if you said, if you know that those three are wrong, then you know the answer is cell walls. But just for the record, prokaryotes or bacteria have cell walls. They're made of different stuff than plant cell walls. Remember, plant cell walls are made of cellulose. These are made usually peptidoglycan, but that's all right. All right, try to answer these. and then we will answer it together. I'll try to zoom in for you. Is that good? Read the problem first, so no one knows, okay? So the problem reads, oxygen depletion, also known as hypoxia, is one result of runoff from over-fertilized lawns or farmland. After heavy rains, nutrient-rich waters enters the local aquatic environment, causing a series of events to occur within an ecosystem. Some events caused by nutrient-rich runoff entering a lake ecosystem are shown. Some of the sequence labels into the blank, or move the sequence labels into the blank boxes to sequence the correct order of events. Well, I wanted first to show you what I had already showed you before, which I showed you at Lake Erie on Lake Erie around Toledo earlier, I want to say a couple years ago. The algae bloom got so bad because of fertilizer runoff from people's lawns and from farms, and also sewage, raw sewage getting dumped into the lake, into Lake Erie, that the intakes for fresh water were clogged and people didn't have fresh water to drink. So that's how bad this stuff can get. And it's not just fertilizer on lawns, it's also city sewers getting dumped. When it rains a lot in this area, when it rains a lot, the rain sewers combine with the toilet sewers, and the poop goes right into Lake Erie. And what you get is it used to get, and it's going to be less and less now because the Cuyahoga County Regional Sewer District has increased your parents' water bills so that they can fix this problem. Anytime that happened, a big rain, you get notices on Lake Erie say, no swimming allowed, E. coli warning. Where did the E. coli come from? People's poop. So it's an issue. But what plants need is fertilizer. That's why people put fertilizer on grass. To do what? To help grass grow. Fertilizer is made of nitrogen and phosphorus. Farmers put it on cornfields and farmland in order to make plants grow. So when we look at what happens though when that runoff from these lands, the rains, falls on here, someone put fertilizer on this to keep it green, it comes in, that fertilizer falls into this pond, what happens to the pond? What grows in that pond? Algae. Plants, or plant-like organisms, right? Photosynthetic organisms grow. Now you would think, wait a minute, that doesn't make sense. How does that make hypoxic conditions? How does that take away oxygen if they're doing photosynthesis? You remember our discussion? Is that it? All right, go ahead.
They start to block the surface of the water.
That's right. That's right. They block the surface of the water, and the plants, the algae underneath them, let me go ahead and draw you a sketch, kind of a side view of it. So if this is the pond, and this is the water surface, there's algae grows on the surface, and normally algae grows on the surface, that's fine, it's normal, it turns a little green, not a problem. But if it gets thick, if there's a lot of algae, why would there be a lot of algae? Well, how could the algae grow so much in just plain water? It wouldn't. It grows when there's water and what? Fertilizer. And that could be poop. Poop is a good fertilizer. It could be anything that has nitrogen and phosphorus allows this algae to grow. But what happens to the algae down here at the bottom? It doesn't get any sunlight because it needs water, which it has. It needs fertilizer, which it has. But what's the other thing it needs to do photosynthesis? The sunlight. So this part of the algae is deeper down in the bed. It can't get sun, so what happens to it? It dies. As it dies, where does it go? It falls to the bottom, right? And layers on the bottom. And what happens on the bottom? What's down there waiting for them? It's actually throughout the pond, but well, fish won't eat it. I mean, some fish will eat some of it, but what happens to dead things? What happens to dead things? decompose. So we have things that decompose them, do we not? If you die, something's going to eat you. It's called mold, fungi, bacteria, right? We call these things decomposers. They break you down. They take you apart. So as the algae dies, the decomposers. Now, do the decomposers do photosynthesis? No, they eat. They're eating dead things. So they're not doing photosynthesis. Here, these guys are doing photosynthesis. They're taking water, right? They're taking sunlight. They're taking CO2, and they're making what? Sugar, right? And oxygen gas. So these guys are making oxygen, no problem. These guys are taking what? The sugar. Where are they getting the sugar from? The dead things. And they're adding to it oxygen. So they're taking oxygen away from the water. And they're making what? Well, ATP for sure, right? And what else? Hopefully everybody says, and not everybody's saying it, so I'm assuming you either know and you don't want to say or you don't know. It's CO2, carbon dioxide. That's what you're breathing out. So that you do this, it's called cellular respiration, you know this. It's cellular respiration. That's what decomposers are doing. They're breaking down the sugar, producing carbon dioxide. So now the poor fish, what happens to the fish? They don't have enough oxygen, and what happens to them? They die. They're dead. And then when they die, what happens to them? They get turned into CO2, they get decomposed, and there's more and more CO2, less and less oxygen, more and more death. And eventually you get what you see here in this diagram, just awfulness, right? By the way, if you look at the breakdown, so you got, this is all kinds of pollution that can happen, but you have fertilizer, seepage, and that can happen on any farm. Whenever you're growing meat, remember we talked about it in the fall. Anytime you're growing meat, what do cows, what do pigs, what do chickens do? They poop. And we talked about the fact that poop, they used to have whole pools that collect the poop so that it doesn't run off. Sometimes it runs off and gets into what? The water supply, and that can cause that eutrophic lake to happen. By the way, it could run off and get into our vegetable gardens next door. If there's a farm next door growing romaine lettuce, then all that poop water is going to go into the romaine lettuce, and you're going to buy it at the store, and then what happens to you? You get sick and might die. Do you all know that there's a romaine lettuce ban right now, right? Have you noticed that? People told you don't buy romaine lettuce because I think it's something like 20 people died from E. coli poisoning. Where'd that come from? The poop from some pigs or chickens or cows. Huh? It's specifically from a region, and I don't know the region. You'll have to do some research on it. I just gave up eating romaine because I don't know where it comes from. Do you know where the romaine lettuce comes from? I don't. So I'm not eating romaine until somebody says it's okay. But that's just me. I'm nervous about eating any lettuce, to be honest, at this point. But anyways, that's eutrophic lakes. So now let's go back to the order, and you tell me what order, and see how easy it is, by the way. Tell me if you don't think it's easy. Which one came first? 4. Wait, the first, which is first?
Oh, that's the bottom.
The bottom one. So number 1 is algal blooms. Now, if they had fertilizer in here, fertilizer runoff, that's what you would put, right? Because that's the first thing that happens. Then what's the second thing that happens?
The second one.
The second one happens, so algae consumes the nutrients and begin to die off. Okay, what happens next? The decomposers break down the algae and increase in number. And what's the last step? They increase oxygen consumption. If it gets bad enough, fish die. Decomposers, this is a positive feedback loop. More fish die, more things die, more decomposers, less oxygen, more things die. It just keeps getting worse and worse until the lake is dead. That means nothing alive in the lake, no animals, because there's no oxygen. That's not good. Okay, we still got some time, so let's do the next problem.